Good afternoon. My name is Arda Halu, and I'm an instructor in medicine at Brigham and Women's Hospital and Harvard Medical School. Today I will talk about our method, MuXTalk, and about detecting and dissecting signaling crosstalk via multilayer networks. Signaling pathways are sequences of biomolecular interactions through which cells respond to their environment. Together, these interactions are often modeled as networks and analyzed using network methods. In the larger network of signaling interactions, the paths of signaling pathways often cross. This phenomenon is called signaling crosstalk, and it occurs when the stimulation of one pathway's receptors results in downstream effects on another pathway. Signaling crosstalk is involved in healthy developmental processes as well as disease processes. Analyzing signaling networks comes with its own set of challenges. First, signaling networks consist of multiple types of concurrent interactions. Second, signaling edges are heavily context dependent. An edge between the same pair of nodes may have different meanings in different pathways. Third, signaling is inherently entangled with transcriptional regulation, which results in various feedback mechanisms, as well as crosstalk. As a means to address all these challenges at the same time, we use multilayer networks, which can represent multiple types of concurrent and context-dependent interactions, both on signaling and regulation levels. In particular, we use the concept of multilinks to characterize signaling pathways, study their crosstalk, and gain insights into the interplay between signaling transduction and transcriptional regulation. Our analytical framework, which we call MuXTalk, briefly consists of the following steps. We first build the multilayer network consisting of signaling and regulatory layers. Central to this multilayer framework is the concept of multilinks. Multilinks count as a unique type of edge every possible combination of interaction types across all layers. We're interested in the statistics of these multilink types. We want to see if they're statistically over or underrepresented in the network. For this purpose, we generate an ensemble of randomized networks and then compare the actual counts to the counts of the randomized instances to calculate the z-scores and empirical p-values for each multilink type. Here's what this workflow results in. For each multilink, the first digit represents the edge type of the signaling layer and the second one the gene regulatory layer. Overall, there are 36 possible multilink types. Plotting the z-scores lets us see which kinds of signaling edges are statistically overrepresented and the extent of this overrepresentation. The overrepresented multilink types offer mechanistic insights about the interplay between signaling and gene regulation. Overall, multilinks of type 0,1 and 0-1, which represent the absence of an edge in the signaling layer and the presence of an edge in the regulatory layer, were significantly underrepresented across all signaling pathways. Inhibition and phosphorylation events were in opposite direction with the regulatory events, whereas dissociation events were in the same direction. In contrast, activation, binding, association, and compound interactions tended to occur without concurrent regulatory interactions. When we look at the overlap between signaling and regulatory edges in the entire network, we find highly significant overlap. On the other hand, the signaling regulatory overlap within each pathway was largely not significant, with a mean z-score of 0.3, suggesting that the signaling regulatory interface is located in the intermediary network region between signaling pathways. Based on this observation, we hypothesized that multilink statistics of edges in the intermediary region between signaling pathways can be harnessed to identify their crosstalk. We devised two complementary versions of MuXTalk, one that relies on direct edges between pathways and one that relies on longer range interactions, or shortest paths. To assess the prediction performance of MuXTalk, we designed a benchmark that utilizes the gold standard crosstalking pathway pairs from a literature-curated crosstalk database. Both versions of MuXTalk surpassed 4 other network-based approaches in both the stochastic and the deterministic version of the benchmark. To discover potentially novel crosstalks, we applied MuXTalk on the entire discovery set of pathway pairs, and we used tailored PubMed queries to assess the validity of the predictions. For both MuXTalk versions, this resulted in over 80% precision for the top 20 predictions. In summary, MuXTalk surpasses current methods in terms of predictive performance, expands the scope of gold standard crosstalk datasets, and offers insights into potential molecular mechanisms leading to crosstalk. I'd like to thank my colleagues at the Brigham for their collaboration and the NIH for funding this project. Thank you for your attention.